Okay, so, a lot of times I'll get asked, how do you apply biological knowledge to kind of real life? And this is just a very kind of great example that I'm going to tell you here, where you can take knowledge from biochemistry, genetics, and apply it to real life scenarios such as exercise, adaptation, if you just have a little bit of foresight and you have the ability to put things together. So what do I mean by this? So there's this concept in biochemistry which describes how enzymes behave, called the Michaelis-Menten analysis or Michaelis-Menten equation. And this would, on the y-axis, be the rate of a chemical reaction, and this would be the concentration of some substance X. Okay, and this is what the curve would usually look like. Okay, so this curve represents the rate or the speed of a chemical reaction as a function of the concentration. And this guy can be two phases. This graph has two phases. One is where it is first order reaction, and the other is where it's second order. Okay, and you might be zero order, and you might be saying, okay, how does this kind of represent an enzyme? Well, here is an enzyme molecule. Okay, an enzyme molecule will bind a specific substrate. Okay, so that would be our X here. Okay, and it will then catalyze it and then make some product Y. Okay. But there is a point where this enzyme is so saturated, meaning it is so filled with substrate, that the rate of that reaction, the speed of how fast that reaction occurs, can no longer be changed by the amount of this substrate I add. And the reason for that is that this enzyme will become saturated with so much substrate that it doesn't matter if I lower the concentration of this, right, increase the concentration of this, the rate will no longer change. Okay, and you can see that it remains a straight line at this point. However, at the very start of this reaction, the rate will change. Okay, now this point, okay, where I hit this kind of plateau on the Michaelis-Menten curve is called the Vmax. Okay, it's called the Vmax, meaning that at this point, my enzyme is completely saturated with substrate. Okay, and if I give it more substrate or less substrate, nothing will happen. It will not catalyze the reaction anymore. Okay, now guess what? How can I apply this to real life? Well, in real life, okay, you guys know that we actually have this process of respiration occurring, right, where in our cells we have glucose being turned into molecules of pyruvate via glycolysis. Okay, and this pyruvate can either go into the mitochondria, all right, to produce ATP, or it can be acted on outside of the mitochondria with this enzyme called lactate dehydrogenase to make lactate. Okay, now imagine this pathway in somebody who is untrained, right, and trained. Now, by trained, I mean somebody who does aerobic exercise training. Okay, so they have adaptations. In somebody who's untrained, okay, guess what? This pathway is more prominent. And you might be asking me, hey, well, why is this pathway more prominent? The reason why in an untrained person this pathway is more prominent is because the Vmax of enzymes involved in this process is 10 times higher than the Vmax of enzymes in the mitochondria. Okay, so that means it takes a longer time to saturate the enzymes involved in this pathway than it does in this pathway. But guess what happens? When you are a trained individual, right, you start turning on genes that are involved in increasing enzymes in mitochondrial respiration. And you know what happens to your Vmax when you start increasing the number of enzymes? Well, guess what? Now your curve is going to look like this. Now you have a new Vmax because you increased the number of enzymes. So guess what? When I told you the Vmax is the point at which the enzyme is saturated with substrate, well, it would only make sense that now that I have a new concentration of enzyme, okay, it would take longer for my enzyme to be saturated, okay? And so this right here, this graph right here, is simply showing us a trained versus an untrained individuals' mitochondrial enzymes. Okay, in an untrained, the Vmax is super low. Okay, meaning that a lot of this pyruvate will end up being shuttled to this lactate pathway, right? And that's why their fatigue is a little bit they reach fatigue easier. Whereas in somebody who is trained, guess what? Their Vmax, their ability to shuttle this pyruvate into this direction is much higher. And guess what? They don't use a lot of this lactate. They don't make a lot of lactate, so they don't get a lot fatigued. Okay, so you see how this easy concept of gene expression in biochemistry can be paired up with this concept of training and how it impacts performance.